no problem. All right. So can able to see that. Okay. All right. Can I stop here now?
During presentation.
Yes,
Yeah, during presentation, can you change it to slideshow mode, sir, so that it will be.
Oh, yes, yeah, I'll do that.
Okay, then.
Right. You just need to, because now I have two monitor here. Okay, you have to tell me like, which one is being highlighting. Sometime sometime it may switch.
Okay.
Yeah.
Okay, within 5 minutes, we'll start the program then, Kati.
Okay. So how?
Shall we start now, ma'am?
Yeah. Kati, shall we start?
Yes, we can start now. No problem.
A very good evening and welcome to one and all for the international webinar series DENTOPHARM 2023 on recent trends on hematopoietic stem cell transplantation for the treatment of congenital disorders. To start now with the program, I would like to welcome our chairperson, Dr. Srinisha ma'am, Registrar Dr. Bhuminathan sir, Principal Dr. Jimson sir, and Dr. Sumati Jones ma'am, Head of the Department of Pharmacology at Sri Balaji Dental College and Hospital. Over to Sumati ma'am to share a few words on today's webinar.
Welcome to second set of DENTOPHARM International Webinar Series. Today we have with us Dr. Karthikeyan. He's a very good friend of mine. He's well versed in stem cell therapy. Today he'll be discussing about hematopoietic stem cell transplantation. We all know that stem cell transplantation is necessary in case of immunodeficiency or malignant diseases. I don't want to waste much time. Go to Ramya again for the short introduction about the speaker. Thank you, Kati, for accepting our invitation.
It's my immense pleasure to welcome the guest speaker of today's webinar, Dr. Karthikeyan Kandasamy, Senior Research Fellow at Yong Lowland School of Medicine at National University of Singapore. He completed his BSc in Chemistry at University of Madras in 1991 and MSc in Environmental Toxicology at ALM Postgraduate Institute of Basic Medical Sciences at University of Madras in 1994. Also pursued Diploma in Industrial Pollution and Control at Annamalai University in 1996. Later pursued his PhD in Biochemistry and Molecular Biology at University of Madras in 2001. In the journey of his career, Sir has received Junior Research Fellowship, Senior Research Fellowship from Lady Tata Memorial Trust in 1996 and 1998. Also received Council of Scientific and Industrial Research from Government of India in 1999. Internationally, Sir has received Abney Scholar in Abney Foundation, USA in 2006. He has mentored more than 35 students from university, polytechnic, and secondary students in Singapore. His expertise are on stem cell biology, tissue engineering, toxicology, models, and cancer biology. We welcome you, sir. We are pleased to receive lecture from you.
Thank you very much. Can I start now?
Yes,
Yes.
sir.
Okay, can I share the screen? Let me share the screen. All right. Can you see the screen now?
Yes, sir.
Okay. All right. Okay, great. Okay. Good evening, everyone. My name is Karthikeyan Kandasamy. I first thank Dr. Sumati Jones and the organizing committee for giving me an opportunity to present my research work here. So here I'll be talking about recent trends on hematopoietic stem cell transplantation for the treatment of congenital disorders. So before going directly into the transplantation strategies, so I'm going to introduce a little bit on stem cells first, then about slowly then introduce new hematopoietic stem cells, then going into the congenital disorders and how these are related, then go to the transplantation. So obviously, when we talk about transplantation, so we'll be also be a little bit talking about the immunological part of the transplantation and slowly get into our research work. Okay, all right. So let's go to the basics. Okay, so you already know about stem cell, just a kind of refreshing for you. The stem cells are undifferentiated cells, are unspecialized cell. So it has two major properties. So one is the self-renewal. So it can generate itself and make more stem cells. And the second one is differentiate into other cell types here. Okay, so depending on the type of cell, whether the stem cell can give rise to multiple cell type or very few cell types here. Okay, so let's look at stem cell types here. So naturally, there are two cell types. So embryonic stem cells and adult stem cells. As the name implies here, embryonic stem cells come from the embryo, and adult stem cells, we already have it. Okay, so check the differentiation. What are the differences here? Embryonic stem cells originate from the embryo, and this is tissue specific. And these cells can give rise to all type of cell, but this is more specific here. For example, this is a pluripotent stem cell, and adult stem cells probably, you can take it as example of hematopoietic stem cell, mesenchymal stem cell. They are all multipotent. They are not pluripotent. And very limited applications are available for the adult stem cell, while the pluripotent stem cell have many multiple applications. Okay, just an example. Okay, if for an example, if my outer layer of skin is peeled off, okay, and what happened? The cells have multiple layers, right? So the layer beneath the peeled off skin then started differentiating, and then it just healed the wound and then cover the whole area here, and then the wound is completely healed up. So that means the layer beneath the skin acts as a stem cell, kind of progenitor cell, and it can completely differentiate and then completely heal the wound. So that means there are stem cells still available in our body. They are just progenitor cell, but can only give rise to specific cell types, not all cell types. For example, these skin cells can give rise to only skin stem cells, but not liver or kidney. Okay, all right. There is also one more cell type, which you may be aware of it. Okay, this is not naturally available. We call them as induced pluripotent stem cells. This induced pluripotent stem cells have many applications been synthetically produced. How this has been done? Okay, let's go a little deeper into that. Okay, you know that there are heart, lungs, different organs are there in our body. Okay, they're functionally different because the genes that control the functions of the organs are different, right? So likewise, if you take the stem cells or the fibroblast, a skin cell, okay, the genes which control these stem cells or the fibroblasts are different. So if we can understand what are the genes are present, so some of the genes which are master regulators of stem cell, if you put into the fibroblast, then the fibroblast can turn into an induced pluripotent stem cells. Okay, so this technology was invented by a Japanese scientist called Shinya Yamanaka, and he won a Nobel Prize in 2012. So, and then the factors, this OCT4 nanoSOX2, okay, these genes are called Yamanaka factors. This is now commercially available, and you can use it to generate normal cells into pluripotent stem cells. All right, let's go to some applications here. So, for example, this is a patient having some genetic disorders, right? And what you can do is from the patient, just take out some of his skin cells or whatever the tissues are easily available without an invasive procedure, and take these cells and then use these Yamanaka factors, convert them into iPSCs. So once you convert them into iPSCs, then what you can do, either if there's any genetic mutation, use gene editing technology to modify it, correct everything, and put back the healthy cells back into the patient. This is one way, it's called cell therapy. Or what you can do with these stem cells, you generate a disease model where you can screen the drugs, whatever the drugs you wanted to play with, you can screen it to get the treatment for the patients. This can be done. So these are some of the medical applications for using iPSC. So other than that, you can also use iPSC for rescuing endangered species. For example, there's a lot of animals getting into the endangered, and in order to rescue it, to maintain the biodiversity is very essential for the human being. So this is one of the applications of iPSC. The other one is a very hot topic now, is a cellular agriculture. Okay, this is one of the hot topic in Singapore and as well as in other growing countries here, because here what they do is, you can take the cells from animals and then you can extract the stem cells, then put into bioreactors, then you can get the meat. So we don't need any large farmlands, no harming and killing of animals, and you can get the quality control products generated in the lab. This is one of the commercially growing food market here. Okay, so for example, like in Singapore, what is happening? We don't have much land, so our raw food supply, we are depending on the outside world. So this kind of technology, Singapore want to look at it so that we can lower the dependency from the other countries. All right, so now let's go to hematopoietic stem cells. Okay, so hematopoietic stem cells are, you can take from the bone marrow. Okay, so this can give rise to many forms of stem cells from two different progenitors. One is a lymphoid progenitor cell, which can give rise to T cells and B cells, and myeloid progenitors, which can give rise to other components of the blood. Okay, the source of HSCs we're looking at. So there are fetal liver and umbilical cord blood are the very good resources for the HSCs, and bone marrow and peripheral blood are the other sources. So the fetal liver, as the name says here, and umbilical cord are related to the pregnancies. Bone marrow and peripheral blood are easily available from the donor, so which can be used for the therapeutic purposes. Okay, and these are some of the genes, genetic disorders, which are associated with hematopoietic stem cells. So we are particularly working on hemoglobinopathies and beta thalassemia. So these are all genetic defects in the newborn babies. So here, now either the genes are deleted or mutated or not expressed, leading to the defective cells, and the defective cells is being reflected in the phenotype. So there are two different methods available. So either you can use a stem cell-based therapy or gene therapy. For stem cell-based therapy, so what you need is a tissue-matched donors needed, and you probably go through the frequent transfusion or boosting required. One of the major negative point here is the immune rejection. Okay, the second one is gene therapy. So this is a hot topic now, and there are gene editing tools are available, the base editors and prime editors, with the help of Cas9 system. So we can use it to correct the gene mutations pinpoint out. So one of the negative points here is the off targets. So sometimes what happens is that when we use these gene editing tools, we may come across maybe off targets affect maybe there's an insertion or deletion of some gene bases into off target area, which may be very deleterious to the person. Okay, so today I'll be talking about stem cell based therapy here. Okay, so far we talk about the just basics of stem cells, its applications, and a little bit on the hematopoietic stem cell. Okay, now I'm going to just go to the transplantation. Why we need the transplantation for these genetic disorders? So we need to see how successfully we can do the transplantation for the treatment of these genetic disorders. So for that, there are some strategies are required. Okay, for stem cell transplantation, what you need is a large volume of cell. For example, you need like up to 1 billion cells per kg of body weight. Okay, and when you inject these stem cells, the receptive bodies need to support the stem cells. And then the other thing is the space availability. So the the injected stem cells need to survive inside the recipient body and need to colonize and produce the lineages for the normal physiology. The other things are, once these cells form colonies and give rise to lineages, these cells, which are being differentiated from these stem cells, need to take part in the physiological process of the body. And one of the major the criteria is we need to have a lower immune rejection mechanism so that the cells will survive and produce and give rise to the differentiated cells. Okay, for example, if you're taking a 50 kilogram person, as a patient, what we need is a 50 billion cells required, which is like huge amount. And in order to support the stem cell, we need to do the preconditioning, like for example, myeloablation. And the person may be having a well-matured immune system, may reject the stem cell, so he will be on immunosuppressive drugs. Okay, so instead, so if we work with the babies which are born or before they are born, it will be easier to transplant because the body weight is so small, and so we don't need large volume of cells. That's one thing. This is a developing body, so this embryo will definitely support the injected stem cells. That's the second thing. And there is plenty of space available for the stem cells to make colonies, and then they can easily get into the required organs, and then can give rise to cells. The major key factor here is there is a T cell selection going on. Say there's an immune development is going on, and then the injected stem cell may be recognized cell. So there will not be any kind of immune reaction to the injected stem cells. Okay, so that's the main concept of having a fetal therapy. And so what we are doing is intrauterine transplantation. Shortly, it is called IUT or IUHSCT, intrauterine hematopoietic stem cell transplantation. All right, having this strategy, so we need to know, once we decided to go for a fetal therapy, we need to know, because the gestation period is 40 weeks in human. All right, so in case of mouse, it's 19 to 21 days. So we need to find an area where we can find a time where we can inject these stem cells. So looking at here, from the starting of the hematopoiesis, so hematopoiesis start from the yolk sac, and there will be a lot of stages it covers in mouse and human. By the end of E12, embryonic day 12, the hematopoietic stem cells migrate to the fetal liver. They stay here for a long time. In case of human, it is around the day of 40. So after that, until the baby is born, it slowly migrate from the fetal liver to the bone marrow. Okay, so at this point, there's a window of opportunity available. If we can inject stem cells at this point, probably the injected stem cells may migrate to the bone marrow and start form colonies and give rise to these progenitor cells and the lineages. Okay, so that's the idea of having intrauterine transplantation of hematopoietic stem cells. So with this method, okay, scientist Alan Flake, he started working on this IUT hematopoietic stem cell transplantation into the first model. He used mice as a model. The first model was very successful. He used allogenic stem cells, fetal liver HSC. It was very successful. The chimerism was more than 10%. Then we want to apply this technique to other models, mouse, canine model, and to non-human primates. It's not very successful. The chimerism level is at a very subtherapeutic level. It's not very high. So what they say, there is a host immune barrier to the allogenic stem cells, what we inject. So there are a lot of studies were going on this technology. Then the reports keep coming up saying that the injected normal stem cells compete with the defective stem cells for the space to achieve the donor chimerism. The other thing they're saying, we need at least 1.8% of initial chimerism to get the required therapeutic level for getting successful transplantation. So if you can inject higher doses to get higher percentage. So what they are saying, okay, there are immune barriers exist for successful transplantation. So one of the main barriers here is the fetal immune system. So overcoming host immune responses is necessary to achieve higher percentage of donor cell chimerism. The other barrier is the trafficked maternal cells. Okay, so what happens here is, so the baby is inside the mother's belly, and there is a two-way bidirectional movement of cell. For example, here, from the baby, there are few cells that is being traveled to the mother circulation, and some cells from the mother traffic to the fetal body here. Okay, so this bidirectional transfer of cell taking place when there is a normal pregnancy going on. So we feel that the this trafficked maternal cell and fetal immune cells, probably they are the major barriers. If we can understand these barriers better, we can tame it so that we can get successful hematopoietic stem cell transplantation. So let me go a little bit onto the maternal trafficked cell. So what is happening here? So this is one of the maybe emerging area. So we call them maternal microchimerism, shortly MMC. So what is happening here is, take a parents here, mother and father, okay? And the mother is having a baby, inside her belly, and there is a traffic going on. That means some cells from the baby is going into the mother. We call them fetal microchimerism. Some cells from the mother go into the fetal body. It's called the maternal microchimerism. So this can happen in normal pregnancy. Okay. So if the mother is bearing the second child, what happened? There are cells from the first child is still circulating in mother body. So it can go to the second child and subsequently to the third child or fourth child. Okay. At the same time, the cells may be passed from the first generation to the second generation to the grandchild. So for example, in my case, I have my mother's cells still circulating in my body, and maybe my grandmother's cells still circulating in my body, maybe my maternal uncle cell. At the same time, if I have my elder brother or elder sister, my elder brothers and elder sisters cells also circulating in my body. Okay, so people are fascinated by this transfer of a small portion of number of cells that has been circulating into the child body from the mother, from the grandmother. Okay, so this generation, this transfer of cell going on from generation after generation, they wanted to study that. They want to coin a term. As we know that now when we study about genes, we call them genome, right? And when we study about proteins, we call them proteome. When we study about microbiology, we said microbiome. So when we want to study this maternal microchimerism, they coin the term microchiome. All right. So what these maternal cells are doing? So when report has been saying there are some beneficial effects and negative effects that are associated with the trafficking of this maternal cell into the fetal body. So probably it is promoting the immune development in the fetal body. Maybe it is also replacing the injured cells. And also probably it is also priming the fetal immune system for tolerance to NEMA. The NEMA means here non-inherited maternal antigens. As you see here, the fetus received 50% of the cells from the mother and 50% of the cells from the father. All right. So there are some 50% of the antigens still not been inherited to the fetus. So probably the whole cell that is coming from the mother trafficking into the fetus, probably it is priming the immune system to tolerance towards the non-inherited maternal antigens. And maybe, of course, there are maybe some negative effects of the trafficking. Probably the trafficked cells may attack the host cells and tissues, and maybe some alloimmune inflammatory responses to the host. So these are some of the properties of the cells that is trafficking from the mother to the fetuses and maybe developing and guiding the fetal immune system. There is also an additional immune help coming from the mother in terms of breastfeeding, in terms of like antibodies can cross through the breastfeeding. Okay, so in all these points telling us that somehow these maternal trafficking cells are involved with fetal immune development. So let's go into the fetal immune development. What is happening here? Okay, so see here, we have the human embryo develop in 40 weeks of gestation. So as early as 4 to 6 weeks of development, you see there's a generation of macrophages taking place, dendritic cells, monocytes coming up at very early stages. Then comes B cells, T cells, then some memory cells keep coming up. So what people are saying that, so this fetal immune system develop as early as possible, and this fetal immune system is active. It's not like very immature, they are still active, but probably during early stage of development, this fetal immune system may be establishing a tolerance at the early stage and maybe giving rise to a functional immune response at the later stage. So maybe there is an active immune system is going on in the fetal environment, and maybe functionally different from the adult. So at the same time, if you're looking at these maternal cells trafficking to the fetal body is taking place as early as 7 weeks of gestation. That means what is these maternal cells are doing at very early stage of this fetal development. So what people are saying, okay, probably this fetal, like this maternal cells probably is priming the fetal immune system for tolerance to anemia, as I told you. At the same time, recent reports are saying probably this maternal trafficking cells may be protecting the child or baby from infections. And the report saying that they could able to characterize this maternal trafficking cell, they isolate it, and then they found the trafficked cells contain T cells, B cells, dendritic cells, and some unclassified cells. So for example, so these dendritic cells, so which circulate into the fetal body and may sample the antigens around it and present it to the T cells. And depending on the type of responses, it may be give rise to an tolerogenic immune responses or immunogenic inflammatory immune responses can happen. Okay, so how do these people detect this, this immunological cell? There's a technique called flow cytometry. For example, the cells having receptors on its cell surface. Okay, for immune cells, okay, these receptors are identified by a molecule called the CD, cluster of differentiation. For immune cells, CD3 is one of the receptor, and subclassified cells, helper T cells, CD4, and cytotoxic cells express CD8 molecule. For B cells, CD19 and CD220 are expressed. For dendritic cell, CD11 molecule is expressed. So these are all proteins are on the surface. So there are antibodies that are commercially available, which are tagged to a fluorescent molecule. So when we incubate the cells with these tagged antibodies, these cells are obviously colored as green fluorescent cell or red fluorescent cell. So once these antibodies are tagged to the cell, we can then put into the flow cytometer, and flow cytometer have a laser pointer which can easily identify the percentage of red cells and blue cells from this one. So for example, here in fetal liver, extract the single cells from the fetal liver and incubate with the colored antibodies, then obviously put into the flow cytometer, and then it is going to give us the percentage of cells. Okay, and how this is going to look like? So the software will generate a dot plots here or histogram. So if you have a pool of cell and if you incubate with CD4 and CD8 antibody, the software is going to tell you out of the whole population, the 48.2% of the cells are CD4 positive cells. So likewise, you can get the percentage of the all immune properties analyzed from the population. Okay, so useful using this technology, and then so we wanted to study the maternal microchimerism. So if we can able to understand this maternal microchimerism, this maternal cells, probably we can term these maternal cells for successful HSC transplantation. So we prepared a model, so where we have the female black mice and white POBC mice, we put them into breeding. Okay, so these mice having a common antigen, CD45.2, is very similar to our human HLA antigens. They also have a unique antigen, H2KD for the POBC and H2KB for the black mice. So the F1 generation will be brown in color, which will be having a double positive for H2KB and H2KD. So if we sacrifice these mice and harvest mononuclear cells and incubate with antibodies, for example, H2KB antibody and different fluorescents, and put into flow cytometry, so you can able to see the fetal cells which are double positive for H2KB and H2KD. At the same time, we see some population of cells which are only positive for H2KB, they will coming from the mother. So these are all the maternal trafficking cells. So if we incubate with more immune antibodies, probably the immune composition of the maternal trafficking cells and fetal cells can be analyzed. Right? All right. So with this one, so we sacrifice the F1 pups from embryonic day E12 to E18 until the 6 months postnatally. And we have the mononuclear cells prepared from different organs. and we characterize for the immune properties here. And we could able to see T cells, B cells, natural killer cells, and dendritic cells in the maternal cells that has been trafficking in the fetal body. Okay, and you can see, so what we did, then we go for hematopoietic stem cell transplantation. So we prepared similarly a breeding black mice versus the bulb C. This is at the day we put breeding is E0 and E14. What we do under anesthesia, so we perform laparotomy and then cut open the belly and take out the, just pull out the fetuses. And by the time we prepare, we already have prepared the donor cells, which is bone marrow mononuclear cell, coming from the maternal side, for example, the same species of black mice, okay, but a little slightly a different antigen in order to differentiate the mother cell versus the donor cells. We also use donor cells coming from the paternal side. Then we prepare the bone marrow, then inject intrahepatically to the fetuses. Then after that, we put back the fetuses back into the mother's belly and close by suture, then allow this suture to heal down, then allow the mother to deliver the babies. Okay, hope you will follow. Okay. So once the babies are born, then we find out what is happening when we do the intervention, what is happening after we put the hematopoietic stem cells into the fetal body. What we found in different organs, we feel there are more trafficking is taking place after we inject stem cells into the fetal body. At the same time, we could able to see these more trafficking cells. We could also classify immune cell that is coming out in the traffic. You could able to see that. There are very selective trafficking is going on. And one of the cell that we could able to see is the dendritic cells here. So what is happening to the donor cell chimerism? So we injected, remember, we injected maternal donor cells and paternal donor cells. When we check the peripheral blood and bone marrow, the father's donor cell, that means the paternal donor cells have higher chimerism than maternal donor cells. Okay, so paternal donor cells perform much better than maternal donor cells. And there is a correlation between the engraftment and maternal fetal dendritic cells level. Okay, so what we thought, okay, if this dendritic cell is trafficking from the mother to the fetus and probably presenting our injected donor cells for an immunogenic response, probably these cells probably control the traffic engraftment of the donor cells. So what we thought, we hypothesized that at the time of IUT, intrauterine transplantation, if we can able to suppress the dendritic cell that is trafficking from the mother to the fetuses, probably we may achieve higher donor chimerism. Okay, so we are looking for a dendritic cell model where we can transiently suppress the dendritic cells in the mother. We found a publication came out in 2002. They have a transgenic mice, and the transgenic mice was created with the CD11C, which is the marker for dendritic cell, under the diphtheria toxin promoter. So what happened when we inject diphtheria toxin, within 24 hours, the dendritic population will go down in the body. Okay, they found just 24 hours later after diphtheria toxin injection, the dendritic cell level go down, stays lower, and then it slowly is coming back to day 6. All other cells remain normal. So using this model, now this model is commercially available in Jackson Tech laboratories. We purchase CD11 TTR mice. Then we put them for breeding with the same bulb C, CD45.2 male mice. The day we put them is E0. So on day 13, pregnant time, we inject diphtheria toxin to lower the dendritic cells. At the same time, for controls, we use saline. So 24 hours later, we prepare donor cell either it is coming from the B6 mice or from the paternal side. We call them paternal IUT, or this one maternal IUT, or completely different donor cell. We call them allogenic IUT, or just a cell line. So we inject the bone marrow MNCs into the fetuses intrahepatically at E40, then put back these pups back into the belly and allow the mom to return. And once the mom delivers those babies, then we sacrifice these babies at few time points, then harvest to see what is the chimerism level and what are the trafficking takes place and immune properties of the cells. Okay. So we harvest it and analyze for donor chimerism and immune response. Okay. So what happens when we inject diphtheria toxin? So just 24 hours later, okay, so the dendritic cell level goes down and remains stay at very low level until day 7. It comes back in some of the like organs like uterus and bone marrow. Okay, we could able to see that just depression in the dendritic cell levels. All right. So this is the plan here. So once at this time, once the baby is born, so we're going to characterize them for immune responses and donor chimerism. So here we need to understand how we analyze the immune responses here. Okay, so what's happening, just a basic here. So this mature dendritic cell, once we inject the donor cells, is going to present it to the naive T cells. So these naive T cells become activated, secrete cytokines, and these cytokines help to make the T cell expansion rapidly. So once this antigen is present, so these cells are called effector T cells. So once these effector T cells neutralize the antigens effectively, the population will rapidly go down, leaving only the memory T cells in the organs. So at the same time, if this immune responses going through the regulatory T cells probably is going to block the effector T cell response, and probably there's a tolerogenic environment is going to be created. Okay, so there is also one more technique we perform when the babies are born. So remember, these F1 pups are already exposed to the donor cells, maternal cells and paternal cells. So we take out the splenocytes from the F1 pups around 2 months old, okay, prepare the splenocytes and incubate again with the donor cell for 72 hours, and then see what kind of immune response you can get it in vitro. So what we can do, we can analyze these immune responses by memory cells, by antibodies which are specific for the effector memory cells, and for regulatory T cells, and for GVHT activated regulatory T cells. We can also analyze the cytokines which are responsible for signaling for the T cell expansion by qPCR analysis. Okay, so let's see here. Now this is a little complicated here. So we have 3 different donor cells we have injected from paternal donor cells, maternal donor cells, and allogenic donor cells. We look at the bone marrow and peripheral blood, and we have 2 systems here. One is a DC depleted environment. One group is DC control environment. Just to see here, when come to the trafficked cells, when we injected allogenic donor cell, there are more traffic that takes place compared to the maternal donor cell injected or paternal donor cells. When come to donor cell chimerism, what we see, there are more chimerism when we inject paternal donor cells. Then comes the maternal donor cells. In case of allogenic donor cell, the chimerism level is microchimery, very, very less percentage here. Okay, so what happened in the group of paternal IUT? So when we see in the bone marrow or peripheral blood, there is no much differences in DC control and DC depleted regarding the immune parameters from the recipients or from the maternal trafficking cells. At the same time, functionally what is happening, so when we incubate the splenocytes from the fetuses with the donor cell antigens, we found that the regulatory T cells are going up when the maternal DC is depleted. So there's a higher CD4 and TD8 regulatory T cell responses when there is a maternal cell depletion takes place. So looking at the maternal IUT in this group, we also found there is no major differences between the DC depleted group and DC control group in the maternal side as well as in the recipient side. Functionally, when we look at it, here also, when we deplete maternal dendritic cell, there's a higher response in terms of regulatory T cells going up when the maternal DC is depleted. All right, so it looks like, so when we inject stem cells into the fetal body, it induces tolerance, probably thus going through a tolerogenic mechanism, inducing regulatory T cells in that scenario. So what we did, when these pups grown up to 2 to 3 months, we again injected boosting with bone marrow MNCs, same type of cells injected into the pups and see what kind of immune reaction we're getting it. So after injection, we didn't do any kind of preconditioning, and we injected bone marrow mononuclear cells intravenously during a retroorbital route. And pups were sacrificed 4 weeks after the boosting. So we look at the organs here, okay, no major differences when we inject either the paternal donor cells or maternal donor cells. We also found there is no antibody generated before and after boosting of these donor cells. So what we found out, no significant differences in immune parameters after boosting. And donor chimerism levels not changed because we didn't do the preconditioning. And there is no antibodies against donor cells observed. So the recipients were tolerant to the semi-allogenic donor cells. So with this periodic analysis, what we found out, okay, there is the immune properties of the maternal donor cells did not change due to disease suppression. And the fetal cells did not show any kind of immune changes due to maternal disease suppression. Functionally, regulatory T cells are involved and subdued cytokine responses when maternal disease suppression is being made. And we don't see any antibodies against donor cells, even after boosting with the same donor cells. So what we did, so we found out only that the tolerogenic environment being produced when we do this IUT. We take the pups at week 0, and then we did RNA sequencing to check whether what kind of gene expression profile and repertoire profile exists when we do the donor cell injection. So here, so we have 4 groups here. So we have control, which is just a normal pregnancy without any intervention. And we have paternal IUT pups, maternal IUT pups. At the same time, we have the DC depleted paternal IUT pups. Once these pups are born at week 0, we sacrifice the pups. And by magnetic sorting and FACS sorting, we separated the maternal trafficking cells and fetal cells and subjected to the RNA sequencing. So this is the transcriptomics data here. And if you look at here, so there are some genes which are common between all the groups and some genes which are unique for each of the groups. But majority of them, most of the genes are towards the immune system development is all common between these groups. So we also look at the repertoire analysis here. So what is happening? So when we inject stem cells, there is a rearrangement of genes going on. For example, in case of the latest issue of coronavirus, so initially, for example, when the Chinese coronavirus came to the spreading, okay, there is an immune response. And after that, there's a Delta variant came out, and then that induced a very drastic damages in the India, right? And after that, there's an Omicron variant. So when an antigen keep modifying, how our immune system encounters, how it prepares and then tackles the situation. So there's a rearrangement of genes going on to specifically target the specific variants. So for example, here, this is the variable region in the antibodies, okay? And these variable regions are very much unique. For T cells, this heterodimeric unit here, alpha and beta, is a combined variable region here. Okay, so what happened? This combined variables, we call them as clonotypes. For example, alpha have a 41 amino acid clonotype and interact with the 55 amino acid beta clonotypes. And if these clones are present in higher amount, we call them as clones. So likewise, maybe a different clones may be formed when we inject donor cells. We want to study this, what kind of clonotypes are present here. So what is happening at the maternal trafficking cell and what is happening at the fetal side? So in the control, maternal trafficking cells, without any kind of intervention or surgery done, the clonotypes are more diverse here. And when we inject the maternal donor cells or paternal donor cells, then the clonotypes become very specific. The diversity is reduced and the clonotype is more specific from the maternal trafficking cell. So when we deplete the dendritic cells, it is going back to the similar to the control cells here. So coming to the fetal environment, okay, the fetal immune system, initially there's less diverse, very few clonotype exist. As soon as we inject maternal donor cells or paternal donor cells, a sudden increase in the diversity, because probably this fetal environment is first time encountering some of the maternal antigen, paternal antigens. That's why the diversity is more. And in the DC depleted condition, there is no much changes here. So when you're looking at the frequencies here, so in uninjected control in the maternal trafficking cell, the higher diversity, okay, number of clonotype is very high. And when we inject maternal cells or paternal cell, okay, the number of clonotypes shrink, but the frequency is increased. The DC depleted type, it again goes back to the control. So in the recipient body, in the uninjected types, the number of clonotypes is very few. As soon as we injected maternal donor cells or paternal donor cells, the number of clones increased, and DC depletion did not affect on the fetal side. So there's almost like kind of like a opposite interaction. So what is happening in the maternal trafficking cell is different from what is happening on the fetal side. So in non-IUD controls have greater diversity in the maternal trafficking cells, but less diverse in fetal side. But when we inject the donor cells perform IUT, the diversity is decreased. Here the diversity is increased. And disease suppression did not have any effect on the fetal type. So in summarize the whole talk, okay, maternal dendritic cell suppression did not alter the trafficking of cells. Probably the maternal traffic is altered by the type of donor cells used. As we see that when we use allogenic donor cells, there are more traffic compared to the paternal cells and maternal cells. So maternal DC suppression did not affect the fetal immune parameters. But you can see there's a functionally regulatory T cells and subdued cytokines levels are going up. Probably it is inducing a tolerance. Okay, so donor cell chimerism is not influenced by maternal disease suppression and looks like they are independent of maternal traffic. And we also found paternal donor cells engraft better than maternal donor cells, and allogenic donor cells are very, very few microchimeric. So this confirms our earlier findings. So when we inject donor cells, this IUT reshapes the clonal diversity in both trafficked cells and recipient maternal cells, recipient cells. So probably if you play with this clonotype diversity, and then if we can use this clonotype, probably we can term this clonotype for successful HSC transplantation. So take home message, if you can term the maternal immune cell, would help fetal immune responses to induce higher tolerance. And the trafficked maternal cells did not prime all NEMA here, because once we inject the maternal donor cells, the diversity is increased. So this maternal trafficking cells is not inducing priming all donor, all antigens into the fetal side. And what is happening here? So in terms of the donor specific and T cell and B cell receptor clonotypes, which can be manipulated to induce tolerance, at the same time can be utilized for successful hematopoietic stem cell transplantation outcomes. Okay, so Some of the findings were published recently in Stem Cell Research and Therapy. And I want to thank our colleagues here and the funding bodies here. This has been funded by Ministry of Health and National Medical Research Council. And here, our boss here, is a research director. And myself and the senior person here, we are working on the mouse model. And Yanti, Julie, and Yvonne are working with the non-human primates monkey model. And we have the new staffs recently joined, and we collaborate to work on this project. Okay. Recently, we celebrated 100 years of our department. And we are, this was last year happened, and then I was proud to be a part of the organizing committee. Okay. I thank you everyone for listening to my talk and happy to have any questions. Thank you.
Thank you, sir, for the wonderful lecture. We'll move on to the question session. Over to Sumati Jones now.
Thank you, Dr. Karthi, for the very good explanation. Actually, the explanation was really clear, and I hope each and every participant must have benefited. Coming to the question, we don't have much question. The explanation was clear right from the basic stem cell to advanced cell research in published in the high impact journal. And your work is really good. Coming to the questions, what will be the limitations as such for this stem cell transplantation therapy?
Okay, good question here. Okay, there are limitations. Okay, so see, looking at here, okay, so we need to find out a good match here for the donor cells. That's one thing. Okay, so when we want to do fetal therapy using intrauterine transplantation. Okay, so here, who is a good donor? So we need to find out. Usually, what is happening here. Mostly the mom is the person who is to donate the bone marrow cells. Okay, the mom is already going through the pregnancy, and so she also need to painfully donate the bone marrow for the fetuses, for the bone marrow for the fetal growth. So this, she's going through the additional thing. So we found out in our cases, we found the paternal cell also is better than the maternal cell. So probably the father can contribute here. So in order to do the fetal intrauterine transplantation, what we need to know is the first, we need to identify the mutation as early as possible. Okay, for example, when the baby is still inside the mother's belly. Okay, so we need to do a non-invasive prenatal diagnostic test to find out the mutations. This is little difficult for the mothers to understand. So this is one of the difficulties the mothers can face. The second thing is how many cells we need to inject. That's one thing. So how good is the immune responses? All this we need to see it. So these are some of the things still kind of like the limiting factors here. And probably if we can term this immune responses, probably we can get the successful transplantation. Hope I answered the question.
Thank you so much. Over to Ramya, the next question.
Sir, are there any recent studies on stem cells from the dental tissues?
Okay. Dental tissues. Okay. Dental tissues, of course, they are one of the good source of stem cells. Okay, people use dental pulp stem cells. That's one thing, and exfoliating. For example, that the baby teeth that is coming out when the adult teeth is going up. So these are the source of cells. At the same time, there are also stem cells that come from the other oral area. You can get it. So people have used these cells, differentiating to different cell type. People have done it. Okay, and this is a rich source of mesenchymal stem cells. People are using, but I never see any studies for the transplantation work for this type of problem, congenital problem. But people use stem cells from the oral tissue and from dental. has been done. A lot of work has been done on that.
Thank you, sir. Thank you, sir, for the knowledgeable lecture briefing on the basics of stem cells, flow cytometry analysis, and role of maternal and paternal donor cells. Sure that our students had a valuable lecture this evening. Hope to have more lectures in the upcoming days, sir. Thank you so much.
Thank you very much. Okay.
Thank you, sir.
All right. So actually, this is my first time I'm giving a webinar like this. Usually, I used to see the audience in the front. So now looking at the monitor, probably I'm got a little stammering. Probably, please pardon me in case of
No, sir. The presentation was wonderful. It was self-explanatory with the images. It was so nice, sir.
Thank you very much.
Thank you, sir. I would also like to thank the institution, Sri Balaji Dental College and Hospital, Dr. Sumati Jones, ma'am, Head of the Department of Pharmacology, for conducting the series of webinar on DENTOPHARM 2023. Principal, Dr. Jimson, sir, and the organizing committee, Dr. Satish, Mr. N.P. Murli, for coordinating for the webinar. Thank you.
Thank you.
Thank you.
Thank you, Dr. Karthik. Thank you so much.
Thank you. So I can leave now, right?